Welcome back to Anatomy and Physiology on Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. In this video, we're going to discuss the functions of the portal triads of the liver, or hepatic portal triads. This is a seemingly complicated topic, but it's really just a conglomerate of a bunch of stuff that we've already studied, and you've probably studied in the context of your anatomy course during the digestive system. So let's actually look at this diagram first over here. So the portal triads are going to be associated with the liver. Now before we get into the portal triad itself, when we look at the liver, we're going to look at a small piece of it, microscopic, and we see that the liver is organized into these regions called lobules. These are hepatic lobules, and notice that they're kind of circular in shape, at least according to this artist's rendition. There can be some variation in that, certainly. And so each one of these, here's 1, 2, 3, and then 4 right here in this image, there are 4 hepatic lobules. Now if we look at this lobule over here, we actually have a piece of it sectioned out, we can see in the center that there's a large central vein. Notice there's a central vein actually in the center of each of these lobules. Okay? We'll talk about what that's for in a minute, but first the structure. And then if we look on the periphery of each of the lobules, kind of going around it in a circle, the periphery has some scattered portal triads. Now let's take a look at this portal triad. The portal triad is a triad, so it's composed of 3 different vessels. The first one is a branch of the bile duct, the second is a branch of the hepatic portal vein, and the third is the branch of the hepatic artery. And then if we notice, interspersed between this portal triad and the central vein, all of these cells in here, these are hepatocytes. Now, really, to understand the interplay with the portal triad vessels, we need to understand about hepatocytes. Now, hepatocytes are really cool cells that have a lot of functions. And I'm just going to go over a few of them that are relevant here. One function of hepatocytes is the synthesis of bile acids. So bile acids are derivatives of cholesterol. They're steroid-like molecules, and these are the main ingredients in bile. Recall that bile is made in the liver. It's stored generally in the gallbladder unless you have gallbladder removed, and then whenever you consume lipids in the diet, the gallbladder squirts that bile into the intestine, the duodenum, so that you can emulsify those fats and aid in digestion. And so the hepatocytes are the main synthesizers of those bile acids. Okay? Another thing that hepatocytes do is they perform a huge array of metabolic transformations. So they're involved in all sorts of metabolic pathways. For example, they perform gluconeogenesis. So at times when you're in the fasting state and your blood glucose falls, they perform gluconeogenesis. And along the same lines, also when you're fasting, they can both store and break down glycogen to release glucose into the blood. They play a major role in lipid metabolism, xenobiotic metabolism, which just means foreign drugs and medications, they metabolize those. So they're a huge metabolic powerhouse type of cell. All right, so now that we understand the functions of hepatocytes, let's see how those functions relate to these vessels within the portal triad. So I've mentioned that these hepatocytes are important in the synthesis of bile acids, which are going to be the main ingredient in bile. And so when these hepatocytes synthesize those bile acids, they're going to shunt those bile acids or dump them into these green vessels, which are the ducts that lead to this branch of the bile duct. And so when these hepatocytes make it, they send it here, and it's basically going to carry all that bile and all its constituents to the gallbladder. And of course, there's a series of ducts that it has to work its way through, but the point is when the hepatocytes make the bile acids and the bile, they're just going to send it through these little vessels, and it's going to go to this branch of the bile duct, which ultimately carries it indirectly to the gallbladder for storage and concentration. Okay? Now let's take a look at these hepatocytes as their functions relate to gluconeogenesis and glycogenolysis. And actually what I want you to notice is that they lie in close juxtaposition to these vessels right here. These are capillaries called hepatic sinusoids. And sinusoids differ from most capillaries in the sense that when you look at the walls of the capillary, there's big gaps in between the cells that make up the wall. And these gaps allow things to kind of just float through. So some things may float from the blood in the sinusoid to these cells, or they could go the reverse direction. It could come from the hepatocyte and go into the blood, and it's just passing through these very large gaps in the walls of the sinusoid. And these sinusoids ultimately are going to connect to the central vein right here. Okay, so let's suppose we're in a state of low blood glucose, a fasting state. We need to elevate blood glucose. Well, the hepatocytes have two things that they can do. One, they can perform gluconeogenesis, where they make new glucose, or they can take stored glycogen and break it down via glycogenolysis, and either one of those processes is going to make more blood glucose. Well, how does it get to the blood? Well, notice that these hepatocytes are in close proximity to the sinusoids. So any glucose they make is just going to go right through the sinusoidal walls, or I should really say the gaps, right? And end up in the blood here. And then it's just going to go to the central vein. And the central vein ultimately takes everything to the venous system, and then it's just in the general circulation. And then that glucose can be utilized by the cells of the body that require it, like skeletal muscle or the brain. Okay? Now, look at this branch of the hepatic portal vein. And we talked about the hepatic portal vein extensively in another video. Remember that when you break down nutrients in the small intestine, they get absorbed by the jejunum, okay, by blood vessels in the jejunum. And these blood vessels ultimately converge into what's called the superior mesenteric vein. So anything that you absorb through the small intestine walls is going to be absorbed into the superior mesenteric vein. So when you have dietary sugar, dietary amino acids, fats are a little bit different, but we'll just say sugars and amino acids, they are absorbed into the superior mesenteric vein. Well, the superior mesenteric vein ultimately drains into the hepatic portal vein. And so then the hepatic portal vein is taken to the lobule right here. And again, it will go from this branch through this vessel right here, and then it can go to the hepatocytes. So when you absorb, let's say, an amino acid, okay, that amino acid goes through the jejunum wall, it gets absorbed into the superior mesenteric vein, and then it goes to the hepatic portal vein, to this branch, let's say, and then through this vessel system right here to the hepatocyte. And the hepatocyte can intake that amino acid and do a variety of things with it. It can synthesize proteins, it can potentially metabolize the amino acid for energy, and the same thing goes for glucose, okay? Except for the fact that you don't make proteins from glucose. You just break down the glucose, or you can build it up into glycogen. And so that's how absorbed materials get to the hepatocytes. They go to the superior mesenteric vein, not shown here, to the hepatic portal vein, and through these sinusoids, they can get directly to the hepatocytes here, okay? And if they're not absorbed by the hepatocytes, if they manage to make it through to the central vein, then they'll just go directly to the venous system and be in the general circulation. And the last thing here is a pretty simple concept. It's that of the hepatic artery, or at least the branch of it. Well, the arteries, we know, carry oxygenated blood. They carry nutrients for the cells that they might need, but oxygen is extremely important. Without oxygen, hepatocytes will probably not survive very well. So oxygen is carried here in the oxygenated blood of the hepatic artery. And then, of course, it's going to come through here, mix with the sinusoids in this capillary network right here. And then the hepatocytes are going to use that oxygen for oxidative phosphorylation and so on and so forth. And so the hepatocytes have all these functions. And the reason why the portal triad, therefore, is important is because it provides an organized way to get things to and from the hepatocytes. And of course, they can go to different regions. The bile duct will ultimately take bile to the gallbladder, whereas the hepatic portal vein brings things in from the digestive system. But ultimately, everything is taken to and from these hepatocytes, and then eventually, things can be moved into the general circulation through the central vein right here. Okay? And one thing I wanted to mention, to be very specific, is if we have any metabolic transformations that occur by these hepatocytes. So let's say we synthesize glucose via gluconeogenesis. When that glucose moves into this blood right here, it's going to go to the central vein. Okay, of the lobule. And the central veins eventually converge into the hepatic vein. And so then the hepatic vein ultimately carries things throughout the venous circulation. And eventually, when that stuff gets back to the heart, it'll be in the arterial circulation, okay, when it's pumped out the aorta. Okay, that's getting into a little bit too much of cardiovascular stuff, but hopefully this video helped you get an understanding of the different pieces of the portal triad, how they relate to both the hepatocytes and the central vein right here. Please make sure to like this video and subscribe to my channel for future videos and notifications. Thank you very much.